Welcome back to the Atma Paradigm, and we're continuing part 3 of our series on life. Today we're looking at psychology drives biology. So this will be an introduction to a new form of biology to some of you, a top-down systems biology, showing how there is coordination in living systems. Now, the good news today is that I won't be bringing any new Sanskrit terms. The bad news is that there may be some biological terms that are unfamiliar to you, but I'll do my best to explain them as they come up. So for this part of the series, I do have to express my gratitude to the input from many of the biologists who are working with the Bhaktivedanta Institute and also with the Science and Philosophy Initiative. They provided a lot of the research and the materials for this talk. And I think that's a great example of our hopes for the Atma Paradigm, which is meant to be a philosophical and scientific framework that acts as a basis for deeper study and research and discoveries. But a special mention to my friend and colleague, Dr. Ryan Bissett, who he received his PhD for research in molecular biology, and he's actually done a lot of the very specific meta-studies and research that has involved the material for today's presentation. So we start with a little recap. Last week we concluded with a simple principle for our understanding of living beings, that life, as we know it here on earth, is the interplay of a conscious entity, the Atma, with a biological body. And we discussed how the inherent characteristics of the Atma find expression to a greater or lesser extent, depending on the type of body that a creature has. And these inherent characteristics of the Atma relate to 3 aspects that we said. We call them Sat, Chit, Ananda. And these represent the primary psychology of the consciousness of the Atma. So Sat is existence, or Sat in terms of how we're looking at it in life, relates to the Atma's nature as non-temporal, outside and unaffected by time, and how this is manifested in life and its sense of survival. The idea that each life form acts to try to perpetuate our period of existence and awareness within this body. And then next is Chit, which relates to the Atma's nature as sentient, aware or conscious, and full of knowledge. And this manifests as the value that living creatures, from top to bottom, place on knowledge. And this is knowledge that we might gain through our senses, something we might find out about the environment, something that might tell us about a threat, something or a special opportunity that there's food available. It's needed for helping us to understand how to respond to events and how to plan and strategize for our best experience. And the third item, Ananda, as it relates to the Atma's nature to be pleasureful and to express connection and love. So even throughout the species, living things seek to establish social and family connections, even across species at times, and we saw some examples of that. And for many, this is the most important feature of what is driving them. Consciousness wants to enjoy, and for that it needs to exist, and for that it needs knowledge. So all these items really culminate in the understanding that we desire pleasure. So the presence of the Atma as the source of consciousness for every individual creature is therefore an aspect of commonality amongst all species types. It's something that unites all life. But that doesn't mean that when embodied, all life exhibits the same level of conscious expression. There's certainly a gradation of awareness, of comprehension, learning, expression, intelligence amongst all species types. However, even with those differences, we saw last week how life seems to share the similar motivations to perpetuate our existence, to learn, to enhance our circumstances, and to maximize favorable experiences whilst in our current body. So each type of species body has a particular set of 2 types of senses. One is perceptive senses. These are the ones we use to acquire information about the world we inhabit. And animals and plants and insects and everything convert this knowledge into plans and strategies for finding food, escaping danger, and also seeking a mate. And these perceptive senses also help these creatures enjoy qualia. They are conscious sentients. They experience qualia, nice things to see, to taste, to smell, to hear, and to feel. And many species seem to have one or more of these senses that offer a more heightened perception and potentially even greater sensual experience than we enjoy within our human body, even though we're so proud of it. And life forms also have another set of senses. These are the action senses. These are the ones by which they move about, or they slither and slide, or walk or crawl, or they run on 2 or 4 or 6 or even 8 legs, or many more. Or they swim with fins, fly with wings. And other senses that they use to manipulate objects and other creatures to attack or defend themselves. Or their sense of communication, by croaks, tweets, mooing, barks, whatever. And, of course, to help them reproduce in all sorts of quite inventive ways. Now, as we said in an earlier talk, there's so many different types of bodies, but each of them has something special to offer the Atma in terms of a positive life experience. So our position is that in our study of life, we must incorporate the presence and the role and the interests of consciousness. And what's been clear from our earlier talks is that nature facilitates life. And as life is the experience and expression of consciousness, nature facilitates consciousness. Basically, nature facilitates life as the experience and expression of consciousness. And this approach offers a much richer understanding, not only of the behavior of life, but also, we're going to suggest, of the biological processes that underpin the workings of living organisms. Now, that isn't the usual approach that's employed by modern naturalists and biologists. They prefer to think of nature as inert, mechanical, chemical processes. What I hope to show today is how that approach is being challenged by a new breed of biology, which has a much more holistic appreciation of what is happening deep within the molecular interactions of our body. There's a couple of biologists who I want to give particular credit to for the work that we're drawing on, and that is Dennis Noble and James Shapiro. And together, they're working with a group who propose a form of what's called systems biology, a term which I'm going to explain, and I will be referring it to for a few times today. Now, I mention this because I believe their work sheds light on the biggest conundrum for biology. There's so many types of bodies, but what determines the particular form that a body develops into? What is the blueprint for our bodies? Where is that blueprint to be found? Now, the common answer is, it's all in the genes. That's what's been taught to us, and that's how most of us really are accustomed to thinking. So we have to start from there and explore that. Now, the story of genes goes back a long way, but I'm going to mention a particular event. In 1953, Francis Crick and James Watson, shown here in this picture, ran down the streets of Cambridge and into a local pub and announced to the world that they discovered the secret of life. And now, DNA was already known as genetics, and heritable traits were already understood. But what Crick and Watson had identified, and we should say with a lot of help from an undercredited colleague, Rosalind Franklin, what they discovered was the structure of the DNA molecule as a double helix, forming the sides of a ladder, and the sequence of nucleotide bases forming the rungs of that twisted ladder. Now, with their discovery, with their insights, we could see what DNA looked like and how it might work. And since then, DNA has attained its A-list celebrity status. It's regarded as the blueprint of our bodies, and all the genes that make up our individual DNA are claimed to define all our traits, our physical features, behavior, attitudes, interests, even beliefs. Now, this isn't without controversy in ethics. Genetic quality and superiority is sometimes cited in racist and supremacist ravings. But our challenge today is really to get the heart of what genes are and what they do, and particularly to consider if genes and DNA as a whole really is the blueprint of life. So, what's a gene? Well, when the structure of DNA was identified, biologists, before they discovered the double helix, biologists used to talk of genes in terms of bits of information that could be passed on from one generation to the next, and which specified the various physical traits of an organism. They didn't know what a gene was chemically or molecularly. They just said there seems to be something which will specify a trait. So we call that a gene. So one gene, a bit of information that would specify one trait. And the idea was that all of an individual's genes added up to being what was called the genotype for that individual. So altogether, a genotype is the set of all the genetic information that would specify all the physical traits. And all the physical traits together make up the phenotype, or all the observable characteristics of that individual being. Okay? So, however, with the discovery of the structure of the DNA molecule, a new idea about the gene emerged, because they could see that there was this ladder with these connections. And this was a fascinating thing about DNA. It had these rungs of the ladder made from just 4 nucleobases. Those are the letters that you see at the bottom of the diagram, opposite one another as the rungs of the ladder. Now, these 4 represent 4 chemicals: adenine, cytosine, guanine, and thymine for DNA. And they're usually known they're just labeled by their letters. And we'll just keep to the letters, which is simple: A, C, G, and T. Now, the interesting thing is that A always connects with T to form a rung, or it could be back to front, T connecting to A. G always connects with C, or C to G, and they form rungs. So you get 4 varieties of rungs in the ladder. But these 4 can be arranged like coded letters in almost unlimited sequences. And so it's noted then, and they looked at this and thought, then a specific stretch of this huge DNA chain seems to be a discrete package of coded information. And that could help to explain how it produces specific protein molecules. So the idea then was such a stretch of DNA coding came to be called a gene. Now, because this seemed to be the active bit of the DNA, the 2 ideas of what a gene was came to be conflated. The first, that a gene produces a physical trait, and the second, that a gene is a single stretch of coded DNA sequence. So putting those together arose the now standard idea that a gene is a stretch of DNA that specifies the hereditary traits of a body. Now, we're going to examine how well that idea stands up during this session. And soon we'll have the promotion of genes, and as a bit of DNA determines our every bit of physical and psychological characteristics. This is how it's grown. And so this idea of the gene fitted well into kind of evolutionary theory and was integrated into the kind of doctrine of the neo-Darwinian modern synthesis. So we talked about the modern synthesis a few talks ago. So there was already the existing ideas that genes were responsible for the physical traits of the body, and that genes could not be changed by physical means, by physical things happening during the life of a creature. Genes were changed only by mutations. Now, with the discovery of the DNA structure, there was a clearer picture of what a gene might look like and what it was in terms of being a sequence of coded information. So now it seemed that we would be able to show a direct causal correlation between every trait of our body and mind and link it with a specific gene. Now, this is not just saying that some genes are important for the function of the body, which is obvious. It's saying that all of the information to specify the form and properties at a macro level of the organism, at our overall body level, that all that information are absolutely contained within the genes of our overall DNA genetic code. So, for the past 60 years, the world of biology has adopted a set of conclusions derived from these presumptions. And these are these presumptions we're going to look at today. They can be summarized as, first, DNA holds all the information, not just part, but it holds all of the information. All you need is DNA. It accounts for everything you see in biology. And this naturally leads to the next belief, that DNA is the sole driver of the actions of the cells. The information in the DNA determines everything else that the cell does. There is no other source of information at work within the cells. That's the thesis. And not only does the DNA tell the individual cells what to do, it also tells the cells how to organize themselves into a complete body. Hence the idea that DNA is, or that it contains, the blueprint of the body. All information to specify its form and attributes can be traced back to the coded information contained within the genes. Now, these conclusions provide an approach to biology which is very clearly bottom-up. It's starting at the bottom level of the genes and saying everything else works up from there. So we have the genes determining the proteins and RNA, which establish the various networks that go on within the cell, which then determine how the subcellular machinery, the bits of chemical machines that exist within the cell, how they do their task, which then says what the cells will do, which then says what the tissue will be, and how the tissues will form into organs and ultimately produce the entire body. That's the thesis. Now, and then once you've got the organism all created from DNA, then natural selection can do its bit, and we can start to see, and there's mutations, and we can start to see how there will be variation of species. So that's the theory. So today we're going to focus on this idea that the biology within our bodies and within all life forms is entirely bottom-up. And based on these conclusions. So we have to focus on these three conclusions and ask ourselves, are they correct? And within that we'll look at the various premises used to justify them, and whether they're right or wrong, or just not completely accurate. So let's explore each of these three conclusions. Now, as I say, we're going to do this based on the challenges raised by many biologists who themselves are evolutionists. They're part of the mainstream academic circle of biology, but they are saying that the old ideas don't stand up to what we now know about DNA and genetics. We're not the 1950s anymore. So much of what I'll be presenting is material which is available in standard biology papers, but also introducing findings and observations by this group who together say that they're calling for a different outlook on biology, which they call the systems biology. So we start with the first conclusion. Does DNA really hold all of the information? Well, let's just see how this works. This is the kind of central dogma, the idea that and it was set up by Francis Crick in 1958, I believe. He said, information flows just one way. It's there in the DNA, and from the information in the DNA, you have a process called translation, where you have the RNA formed. or transcription rather, from DNA to RNA's transcription. And the RNA then attracts the chemical bits to it and assembles amino acids, which form into chains and form proteins and polypeptides, all thanks to the information from DNA. So DNA to RNA first, and then from RNA they start to produce all the proteins, and that's the translation of the information from RNA into proteins. So that's the one-way system that is a keystone of the central dogma. Now, is it really accurate just to think of a gene as a simple stretch of a DNA molecule? Now, the whole idea of using a gene is to set up a stretch of RNA that can be used to form a particular protein. But here's how the process goes. The cell takes a stretch of DNA and forms what's called precursor messenger RNA, as in our diagram. Now, the bits that are of interest to the cell in forming what it needs for proteins are called exons. It's not all of this long stretch of DNA, it's only specific parts, and they're labeled 1, 2, 3, 4, and 5. And they're separated by what are called introns. So the cell has to chop off the introns, get rid of them, because it doesn't need them. They're not important or useful, and maybe even unhelpful to its purpose. Now, then it's got these exons 1, 2, 3, 4, 5, but it might decide, no, actually I don't really want 3 to be included. It may be a very nice bit of genetic information, but it's not what I'm looking for at the moment. So it drops that out and stitches just the others together to form 1, 2, 4, and 5. And this turns the precursor mRNA to mature messenger RNA, using only 1, 2, 4, and 5. Now you could ask, well, who decided that? It wasn't the DNA molecule. The DNA molecule was just the whole bit. So there's no information within the DNA molecule that specifies this choice of how the cell has chosen the exons it wants in order to form the RNA. And it wasn't the only way it could have done it. They've seen in various pathways that there are alternative ways that the cells might have dealt with that. For instance, instead of using just 1, 2, 4, and 5, it could have gone 1, 2, 3, 5 and stuck those together. So it dropped 4 and put the others into its RNA. So you're forming different forms of RNA created by the cell machinery from the same bit of genetic material. Now, the function of each of these, what are called isoforms of RNA, is very significant. It makes a really big difference which exons are included. It really changes the nature of the protein that's going to be produced and what chemical processes that's going to play a part in. So this evidence indicates that DNA itself is not the king. Yep, of course it contains really important information, but the usefulness of that information is how it is gathered, how it is edited and organized. It's like you writing an essay or a thesis. All the information you want is in the encyclopedia. You can go to different parts, or the internet, of course. But how you gather that and weave it together into a coherent composition, that's what counts. Or an example for musicians, think of a piano. Each key of the instrument is like a code. It encodes for a note. Press it, bing, up comes a note. So all the notes needed for all the music, say Tchaikovsky's First Piano Concerto, they're all there. But the piano doesn't and can't play or compose that. So something else is using the information that is encoded within the piano to decide how the tune comes out. Similarly, this is an indication that something is organizing DNA, not necessarily the other way around. And here's another example. When the two strands of the double helix separate, and that's kind of going from the left hand of this diagram to the right, and it forms two new strands, the daughter strands. Well, when that happens, there's an occasional blip, a mistake, like the one circled here in red. Somehow a guanine molecule has become paired with a T or a thymine molecule. Oh, this is disaster. This completely disrupts the coding and the ability of this stretch of DNA to do anything useful. So something has to be done about it. And errors like this happen at the rate of 1 in every 10,000 base pairs. Now, 1 in 10,000 doesn't seem like a lot perhaps, but when you consider the number of base pairs which exist within a DNA chain, then it ends up as an awful lot of errors, during one single act of replication. So help is on hand. The local error correction technical team is ready to step in, so it quickly sees the mistake and reorganizes the connection. It concludes that the guanine, that's the one that was meant to be in the right place. The mistake is the thymine. So let's get rid of the T and insert a proper C in there, a cytosine molecule. Job done. Now, the efficiency of this, it reduces the potential error to 1 in 10 billion. So what that means, that for every 1 million errors that occur, 999,999 are corrected, i.e. only 1 in a million slips through. That's an amazing efficiency. Now, why have I entitled these slides, DNA does not repair itself? It seems to have done a very good job in repairing itself. Well, the answer is because the machinery, which is a 3-step process for the correcting, is not part of the DNA molecule. It's not coded by the DNA molecule. It's part of the machinery of the cell. The cell is correcting the DNA, not the other way around. So, I think we've got a doubling up of my slides here. It can happen. This is showing DNA transcription into RNA and then into the arrangement of protein. But the reason I mention it again is because I want to look at another aspect of how the cell affects the genetic information, or particularly how that genetic information is used. And that looks at the subject of epigenetics. Now, this is a fairly new branch of biology, probably really only came up in this century. And epigenetics involves the modification of gene expression rather than the alteration of the genetic code itself. So, epigenetic effects change how the DNA information is available or how it is used without changing the sequence of the bases. So in our little diagram, you see the ladder, and we've got the bases linking up GC, CG, TA, GC, AT, and so on from left to right. But added to the cytosine molecule is what's called a methyl group. It's a particular chemical which has a methyl CH3 ending to it, which can link in with the cytosine. And wherever this methyl then is connected, it affects the unfolding and the contraction of the DNA molecule. Now, what this effectively does is that the gene that might be very useful and very important for whatever it's supposed to do can be turned off by attaching a bunch of methyl molecules to the side of the chain. So they can the cell adds these. These are not coming from the DNA, they're not specified by the DNA, they're specified by the cell. The cell seems to be deciding, I am going to attach these markers to the C bases within the ladder and effectively turn off that particular stretch of DNA from acting as a gene. It will not be used for coding RNA and proteins. Now that's pretty amazing that this is an example of how the information is arising from the cell, and the cell is saying, here's how I want to use the genetic information within the DNA, not just now, but in the future. So that information, that potency, if you like, is coming from the cell itself. It's doing it to fulfill the functions and purposes of the cell. It is not something defined by the DNA. So this is why the system biologists say, look, you've got to look at this and say, you know, it's not DNA that's driving everything. The cell itself, you know, you don't want to be too anthropomorphic about it, but the cell seems to be making its own decisions about what it is trying to achieve. And it is saying, no, I'm going to switch these on and off. Now, this has real significance, you know, for all of us. Because let me just show you a couple of examples how this works. Here is some recent studies that have demonstrated, this one's from Harvard Magazine, have demonstrated that a woman who smokes while pregnant may induce epigenetic changes in 3 generations. Herself as the mother, her daughter, to be yet unborn, you can see the that's the red baby which becomes the red lady, and her granddaughter. So this is interesting because what it's saying is that the activity of smoking is sending signals to her cells to vary how the DNA code is being used by those cells. And also, it's been passed on. And that breaches a long-standing assumption of the modern synthesis that physical activities such as behavior or the environment can never be passed on in the germline to future generations. Remember the refutation of Lamarck? Well, now with epigenetics, it looks like they can be passed on. Now, of course, this is an example of a person inhaling kind of noxious chemicals into the body. But here are a couple of examples of non-toxic behavior. In the mammalian brain development study, they showed how the behavior of increased grooming by primate parents of their offspring had several effects. One, it increased the lifespan of the children. Nice. It introduced epigenetic markers in the brain cells. It seemed to be connected with emotion, that by encouraging the particular emotion which is a pleasurable and caring emotion, it was affecting the DNA within the brain cells. Somehow or other, the activity was placing these metal markers onto the DNA chain, turning genes on and off. So activity and emotion influenced the way genetic information would be used within those primates. And it was hereditary. It was passed on to the children who also developed similar habits. Now, you might say, well, but maybe this was just learned behavior. Well, here's another example using mice that was done in Emory University. What they did was they exposed male mice to a non-toxic kind of sweet-smelling chemical. So the mice would smell this thing, but at the same time, they would get an electric shock within their foot. And that hurt. Ow. After just 15 experiences that they had over 3 days, these male mice developed a serious fear of the sweet smell. They froze whenever that smell was introduced to them afterwards, even though they weren't getting a shock, because they associated pain with that smell. So later on, they were allowed to mix with some female mice who hadn't been exposed to this experiment. And then they looked at the children that these mice had, and they found that the children also had retained fear of that particular smell. So the indication here is that emotional experience is enough to trigger cell activity, to vary the use of your genetic code to the extent that it can be passed on to your children. So when we come back to our first question, does DNA hold all the information? No. DNA information is corrected, it's augmented, and what exists of it is organized and used selectively, and it is used by the functions and purposes of the cell. Now, we're not saying DNA isn't vital or really, really important. It is, but it's important the way a library is important. Now, a library is continually updated in its content. So is our genetic material. A library doesn't tell the public what to do. The public decide how they want to use the library. When they want to access it, what do they want to look up? What information do they need? How do they want to use it? So the library exists to serve the purposes of the users. And in the same way, the systems biologists say our DNA library serves the purposes of our cells, not the other way round. And this sort of addresses our second point. Does DNA really control cell activity? Because all the evidence that we've looked at so far indicates that DNA does not determine how its own information is used by the cell. Rather, the cell seems to have its own agenda in how it uses the genetic information. Now, here are some other things that happen within the cell where DNA does not contain the information to specify the overall activities of the cell. This is a little video I'd like to show you, and what it is, it indicates the process of mitosis, which is what's happening in one particular cell. You're just looking at one cell, but now it is organizing itself to divide into two. So for this process, as the chromosomes holding all the DNA start to arrange themselves into two neat groups. Bit by bit, the strands of chromosomes gravitate to one side or other until there is exactly the same copies of each on both sides of one cell. Then at that point, the cell membrane starts to close over and thus makes a copy of itself. Two cells, each with identical sets of chromosomes. Now, it may be possible to describe how this is happening, at least in general terms, but why is it happening? Where is the information that says to the cell, now divide and here's how to do it? What is manipulating the chromosomes? This is still the debate. But one thing is clear, whatever is the cause, it has not been identified as information existing within the DNA strand itself. It seems to be a function of the cell's own mechanisms, operating as a system, an action initiated and coordinated by the systems of the cell. So, one response to this conundrum by molecular biologists who are wedded to kind of the old ideas is to say, okay, yeah, right. But, the machinery of the cell and all this incredibly complex functions that are going on, it's all to do with feedback loops. And all these feedback loops are, of course, set up by the proteins. And, of course, the proteins were produced by the DNA molecule. So DNA ultimately is responsible for all of this. Now, that's the standard story, but it doesn't satisfy all biologists when they really look into it. Two reasons. One is that the offspring of DNA coding, i.e. proteins, have relatively low information content. And they do not contain the information to specify the system's activity. They are nowhere near as complex as the other bits of machinery that form the functions of the cell. Now, so DNA does not produce the membrane of the cell and how it works, nor does it produce the various organelles. When we talk about organelles, we're saying, I've got this little diagram here which shows some of the more important bits. It's very simplistic, but it's showing the different bits of biochemical machinery which are at work within the cell to keep it alive, to keep it functioning and healthy. And all these bits of machinery kind of in different colors and different shapes, they're the various organelles of the cell. These are specialized subunits of the cell with each with their specific functions. And they are far more complex than the basic proteins that DNA produces. DNA plays no part in specifying the form and construction of any of these organelles. So how can we claim that DNA is responsible for their exact functioning in all respects? Well, now, of course, then people say, well, the cell and this machinery already exists. We know the DNA doesn't produce them. It just replicates itself. But the cell that it's in already existed because it was part of a previous body and somehow or other has been produced like that. So you kind of go back into the antiquity of time for the cells, saying that all this machinery must have been available way back in the past. It's a chicken and egg infinite regress, but it's a regress that doesn't and cannot start with DNA creating the first organelles. Because you remember, we looked at this in talk number 2 of Life series. We mentioned how the evolutionary biologists suggest that we have to speculate that the first living cell was the chance simultaneous occurrence of 3 parts of the cell. And those 3 parts were the cell machinery, the membrane, and the DNA. And we had to speculate that by chance all 3 happened together. Why? Because DNA on its own does not and cannot produce the organelles nor the membrane needed for the cell to function. In fact, DNA does nothing on its own. It's useful only in the context of a fully functioning cell. So to say that it is all part, that whatever is happening in the machinery of the cell is all specified by the DNA is, short of truthful, let's put it that way. Now here's another conundrum for DNA, and the idea that it does it all. There are 37 trillion cells in your human body. 30, I've counted them all. Check, you don't have to, trust me on it. There's 37 trillion. And those 37 trillion are arranged in there's just over 200 different types of cells that your body uses. This picture just shows a few of them. However, they all start off the same, identical. Now, when a sperm fertilizes an egg, then it forms what's called a zygote. The zygote then divides many times by mitosis, which is the cell splitting that we saw in the video, on its way to form an embryo. Here's the process. We start with our single zygote. It's been fertilized. Now it divides into 2 cells. Both are identical and both have the same DNA, same behavior. Okay? Indistinguishable. Then these 2 cells each split into 2 other cells. Now we have 4. Again, all identical. And then these 4 split again to produce 8 cells, still all identical. They are still considered embryonic stem cells because they each contain the DNA that was there in the original one. Now, at this point of 8, something strange happens. It's almost like the cells decide to convene a meeting, a committee, to discuss the future. And their point together is that as they discuss, if we keep on going like this, where we're all the same, then the body is going to end up as a formless blob of tissue, just made of all the same cells. There won't be any limbs or any other organs produced. So we can't continue like this. We've got to get organized. We're going to need some muscle tissue. So any volunteers? Who's going to be the blood cells? Anyone wants to take on the brain cells? And and they kind of decide that each of them is going to be something different. And that's what happens. At the next split, they're not the same. They've changed from being totipotent, i.e. that they could produce any of the other cells, to be having to being partially specialized, or what's called pluripotent, with the ability to further specialize. And in this way, these 16 end up as 200 types of cells that are needed for the body. Now, the big questions are, why do these cells decide to specialize? And more difficult, how do they know what cell each should become? There's a lot of big questions here. Now, there are some answers that are given to this. The problem is that they're all genetically identical at the beginning. So what makes the difference in how they use that same information? And why it cannot be the information within the DNA itself, because they're DNA identical. And the answer that's given is not to invoke DNA, but this is the standard one that's given at schools and universities. And it's a kind of mishmash of several thoughts put together. The first is that there might be some kind of polarization of the zygote, the cell when it first became fertilized, thanks to the direction from which the original cell was fertilized. And when that zygote divides, it contains a batch of cells. When the zygote then implants itself on the wall of the womb or uterus, that batch of cells is then called a blastocyst. So what they then say is that the position of a particular cell within the blastocyst, as in this diagram, kind of determines what type of cell it's going to become. So the idea is that the cells on the outer part would maybe they become skin, and the bits on the inner part maybe placenta, and the middle bit becomes organs, and maybe at one end becomes the head. Now that doesn't seem particularly precise. And in order for this to work, obviously there has to be some chemical differentiation within the cells. So what is proposed for that is the idea of that there are protein gradients, that there are different concentrations of proteins within a cell. So when it splits, there's, as in this one, the picture to your there, the top cell has all the proteins, the little red dots, over to the left-hand side. So when it splits, the daughters, one has one type of protein, and the other has less of that type of protein. And so this may explain why they kind of go off in their different directions. Ooh, that doesn't sound very precise either. So why is these gradients working, and how does it decide how much of particular proteins go off in which direction? So is this location or the orientation or the proximity of a particular cell, is that precise enough a determinant that it can exactly ensure that a particular set of gene expressions take place within that cell in order to determine what it's to become? Now, for those who have been trained on this, you should know that the original biologist behind this was a guy called Lewis Wolpert. And he admitted in 2011 that actually there's no good evidence for the quantitative aspects of any of the gradients just described, or for the details how they're set up. Basically, he admitted it was a nice idea, but he has nothing really to justify it. So really, these ideas do not contain anything like the precise information that is needed to ensure the correct differentiation of each particular cell in the correct quantity, in the correct locality, and of course in coordination with similar cells and with other cells. So how will this ever translate as an efficient system that will define the setup of 37 trillion cells with 200 types of cells operating within all of the distinct yet interrelated systems that make up our bodies? So each of these kind of ideas and explanations raise far more questions about information and the organization of the cells than they solve. In fact, they make the process of cell differentiation and the development of the embryo appear even more haphazard than we might even have imagined. But the key point that I want to point out here for this part of our analysis is that none of these ideas to explain cell differentiation mention DNA as the driver. There's no suggestion that the cell activity in terms of differentiation or embryogenesis this process seems to be directly related to DNA. So it therefore seems that the information content of DNA within our cells is not sufficient to determine the processes of mitosis and cell differentiation. So for our second question, does DNA really control cell activity? The answer doesn't seem to be yes, it's no. DNA does not directly specify the production of the various organelles and parts of the cellular machinery that the cell has in order to go about its different functions, nor has anyone identified how the information encoded within DNA plays a role in specifying the processes of cell division and differentiation. Now we go on to our third question. Does DNA really determine morphology, the overall form of the body, with its heads and limbs and all the other bits inside us, all in their respective positions? Now, we've got to note that the evolutionary idea does not suggest that the body is designed in a particular way or that it has to be a particular shape. Rather, the idea is that the shape that we're in and that all creatures are in has somehow or other been produced by whatever processes the body's chemical chemicals decide on, or whichever they produced. But it is then being refined over eons of time through evolutionary changes, and that's why you are the shape you are at the moment. And all that information, thanks to evolution and genetics, is now all embedded within our genes. Now, even so, even however the form might have developed over time, regardless of that, this proposition still cannot establish that the template for the form of our current body exists within our DNA. Now, I agree with the systems biologists on this point, that it is hard to substantiate that the template for the body exists within the DNA. In the light of everything we have just established, there is no information within DNA to specify the particular types of cells of our body and how they are used. There's nothing within the DNA chain that specifies how cells will form into tissues and where the form of the organ that the tissue then needs to take is specified. There's nothing there. There's nothing that says how the organ fits in with a particular system. Here we have a heart trying to fit within the cardiocircular system, and how that works. Where's the form of that? And there is nothing for the form of the body. I remember reading a paper in which one biologist was discussing this process of embryology from the top little blue circle, which is the zygote, through the blastocyst, through the development, in embryology, up to the fetus. And he was wondering how the form of the body was everything intact and in place. He just thought, this is an amazing wonder. But he said, it's all thanks to chance. Chance produces it. Every time a body is produced from any species and it forms the way it seems to need to, then that is a wonder of chance. But of course, chance did it. Why? Because we see that the universe is formed by chance. So this is a wonderful symmetry, that every biological body is also produced by chance. Now, I've expressed my views on the explanatory value that I put to those sort of ideas. And as a philosopher involved with the philosophy of science, I would have to question that sort of attitude and dissatisfaction with that sort of level of explanation. Now, rather, I'd tell you that today, that so far, to this day, there isn't a fully developed theory of how morphology, i.e. the form of the body, is determined at the genetic or the cellular level. So, how are we likely to find the information within DNA that can therefore specify the overall form of the body? It's not in our genes, because that's not what genes do. We talk very casually about genes and physical traits and say that they're even personality traits. And every week you may hear scientists have now discovered that genes for this, that and the other. That's starting to wane. That was certainly a trend some time ago. Starting to wane because people realize it's not as simple as that. Genes do a lot, but they don't specify the actual form of an organ or a limb or a whole body. Even the ones that you might think are obvious, genes for height, for instance. No, you can't find a gene for height in your body. They're not connected with a specific singular group of genes. We don't even know how genes specify the length of your arms. Why are your two arms the same length? That is a complete conundrum. So, shown here, I've got a little petri dish with an experiment that was carried out on yeast. Experiments were done to knock out one gene at a time to see what the physiological effect was. And out of 6,000 genes here in the yeast that were removed, 80% had no effect on form or function. So there's not a correlation between one gene and another. And knocking out genes can affect things that not being produced. It stops things produced. It changes, because we looked at that in our study on mutation. So it's not that they have the template for the form that the limb or something is going to take. It's that they can stop the chemical processes that enable that limb to be produced. So here's an example which challenges how DNA produces form. And it shows how the body doesn't rely on particular genes in order to produce the form that it wants for its body. Now, the example is E. coli, Escherichia coli, which is a bacteria. And it swims around and it's got this lovely tail system, it's flagella. Now, some scientists, instead of cutting off the tail physically, like our guy who chopped off the tails of the mice last week, rather the scientists thought, no, we've got to be smarter. We've got to knock out the gene involved in the production of the flagella, of the tail. So they did. They got into one of these E. coli and they knocked out that gene, and then from that tailless E. coli they produced a whole batch of other tailless E. coli bacteria. So when these bacteria, tailless as they are in this picture, were in a particular medium where there was enough nutrients within that medium, they seemed to be content enough. There was no change, they just carried on tailless. But then they put the E. coli into an environment where they were starved of nutrients. But they were starved in the spot where they were. However, the E. coli the bacteria could sense that there was food nearby. And they wanted it, but they couldn't get to it. Why? Because they didn't have a flagella. They couldn't move. That was their limbs. They were just this little cell without any limbs. That thing that they relied on to move around to find their food had gone. Now, it wasn't a problem when the food was everywhere, but now that they needed to get to the food, they had a problem. So what did they do? They started growing a tail, and a usable one. Now, these, don't just think of E. coli as a very simplistic little kind of blob. No, flagella tails are not just a hairy appendage. There's a lot of moving parts, including a complex motor system to make them functional. And somehow or other, the E. coli managed to reproduce a workable tail for itself without using the gene that was supposed to be the one that determined it. So without using that gene, they used a completely different system, different bit of genetic information which they basically bodged together to form different proteins using a different chemical pathway, and generated a tail for themselves. It wasn't perfect, but it was functional. Now, this is just one example of how an organism seeks to reassert its original form. But in this case, it's doing it without relying on any of the genes that were supposed to be critical for that form. Now, if the organism can somehow recreate a physical feature without particular genes, what information is it drawing on? Where is it getting it from? Is there some other template for the form of the body at work that isn't DNA coding? Now, hence, the premises and the conclusions that make up the so-called modern synthesis don't hold. Our three questions. DNA does not hold all the information. It's not the king, it's a library. It doesn't have all the information. It is not the sole source of that information used to specify how the cell operates. And it doesn't contain the information used by the cell on how that library is accessed, corrected, and modified. It does not drive the actions of the cells. It is to be used to produce templates for the proteins that the cell wants for its own purposes. And it does not contain the blueprint of the body. It is not the sole source of specification for the body. It doesn't have the morphology that the body has. Its role is as a tool for the cell. It doesn't contain anything like the information that would determine the form of a body. Now, the evidence that biologists are amassing is that the conclusion is we're under an unfortunate misconception since the 1950s. This is not a bottom-up process. That's why the systems biologists are pressing us to update biology now and realize that we're essentially dealing with a top-down process. Now, the way we're going to start at the bottom, but show that it's a top-down process. So they propose that the reason certain genes are used in the way they are is because they are satisfying the needs to produce certain proteins and certain RNAs. But why are those proteins and RNAs needed? Well, they're needed in order to satisfy the networks that use them. And why do these networks need these proteins in this way? Well, that's because the subcellular machinery, the organelles of the cell and its systems, need those networks in order to do the things that it is trying to do. Yeah, but why do they need it? Well, that's because it's to satisfy the purposes of the cell itself. So this is a top-down imperative at work, where the needs of the cell feed down and influence the operation of all levels beneath it. This is a revolutionary kind of view of biology. But it doesn't stop at the cells. The cell itself is playing a part just to facilitate the formation of tissue. It's trying to be something. It's trying to be a particular type of tissue material. Yeah, but why does it want to be a particular type of tissue material? Well, that's so that that tissue is available to be used to form organs. And, well, why are the organs needed? Well, that seems to be needed in order to play their part within the systems that altogether make up the entire organisms. So, from the higher levels, there's purpose-driven requirements that determine how a system at a lower level will behave. And specifically, it defines that the lower level acts in such a way that it can really only be understood as intended to fulfill the needs of the higher levels, ultimately fulfilling the purpose of the overall organism. Now, according to the systems biologists, this kind of top-down process for living organisms can be summarized like this. And this is the bit that I think you can take away. That there's the organism, the cells, and the genes. And we just keep it to those three aspects. So we have, from the organism, that level triggers the cell signaling. The form of something that is happening at the organism level, at the macro scale of the whole body, that is sending triggers to all the cells, getting them to act in a particular way. And then the cells are sending information to the genes. So in this way, the organism is controlling the action and the use of the genes. Now, this sounds teleological, i.e. some kind of design for a purpose. But this isn't coming from, say, creationists or someone who's got an agenda in any way like that. These are hard-bitten biologists. They're just trying to be true to the evidence that they see. The DNA is not the thing that specifies everything. You know, that's just an illusion we've been fed. Nor does the evidence suggest that it's a bottom-up process, that the genes just feed up and produce everything. No. Of course, they do face the question in top-down biology, well, is the organism the highest level? And if so, where is the template for the form of the body to be found at the organism level? Now, these biologists suggest that there is even a higher level system beyond the organism. They suggest it's to do with the environment, the ecosystem, and the social systems maybe that organism is a part of. Together, this bigger system they refer to as the holobiont. All the influences that come to be factors on the organism. So we would agree to the point that factors such as environment and the challenges that presents will affect an individual organism's behavior and psychology. And we agree that there is even evidence that this has a bearing on how it uses its genes. That's okay. But here is where the Atma Paradigm has a little disagreement with the systems biologists. We don't find any specifying power within the holobiont, i.e. there's nothing in the ecosystem of the environment that says things should be a certain way for an organism. The environment will have an impact on what food's available, what threats are available, what's happening, what the weather is like. It will have a lot of influence on the nature of what a body is within that niche of the environment. That's true. But there's no specifying power within the environment that says you have to be this shape in this way and have these limbs in order to deal with this environment. That's the flaw that is there in this suggestion. So we want to help the systems biologists because we're proposing that there is something to help them. There is something at the organism level that they can draw on to complete their thesis. The idea is that the holobiont, the environment, does impact organism psychologically. Organisms are sentient life forms, and we've demonstrated that in earlier talks. They experience and express their will to enhance their existence. So the environment may place a stress in the form of a threat, or it offers opportunity for enhanced experience. But it is the conscious nature of the organism that then decides how it wishes to respond to those threats or opportunities. Let me give you a couple of examples. This is the Alaskan stickleback. Now, they've seen how sticklebacks with different diets have developed different bodily features. Some of them, if they're in brackish water or in fresh water, they have a different diet, a different medium that they're enjoying. It has an effect on their bodily features. There's thickening of scales, or which requires genetic change. So the many of the biologists that I mentioned in the previous one, the developmental biologist, would point out that somehow or other the psychological intent to do with behavior, food, and environment within an individual or group changes the use of the individual's genes. This manifests as plastic changes in the body, which can be fixed and transmitted to the next generation. Now, this is really interesting. It means that psychology is changing the genetic use. It's changing the form of the body to a degree. It's no help for macroevolution. These are still sticklebacks. They're just a variety within the species. You put them in a different situation, things can switch back. So it's no help for speciation, but it does indicate that the life interests of a creature can determine that creature's alternative use of its own genetic code. As I say, not evidence for Darwinian evolution. This is evidence that of an organism changing the use of its genetic information. Not by random mutations, but by its own intention. And the stickleback isn't alone in this. Here is, we're going to look at the blue mussels on the top left. Now, these are found on the Atlantic near New England. However, there was an invasive species, the Asian shore crab, was introduced to that area, and it was found that where the Asian shore crab was, they found that mussels developed thicker shells to stop the crabs eating them. Now, others don't. Now, this wasn't a process of natural selection. This was a genetic change that the mussels were doing. It was costly to them because in order to produce this extra shell material, they have to use a lot of energy. So they were doing it to protect themselves. So again, conscious experience, intention, and the will to survive influence the use of genetic material for that particular organism. So consciousness may be the drive of genetics and biology, not the other way around. Environmental pressures are real, but they're not specific in the information they give. What they do is to help initiate a response by the conscious entity present within the creature, and so the environment evokes the role of consciousness in terms of psychological intention, which has an impact on its use of its own genetics. So all the environment can do is to pose a threat or an opportunity to the organism. Is there something that wants to attack this creature and devour it? Or is there some opportunity to gain some extra food, a mate, or comfort, or even fun? So the environment has an impact only because it elicits a response or a desire within the mind of an organism. So our question is, is this psychological intention? Is it that which it is generated within the consciousness of the organism that forms the purpose-driven imperative that the systems biology need at the organism level in order to complete their thesis? So this may account for the ability of the organism as a whole, if we include the agency of the Atma, to coordinate the functions of the chemistry of the body. Now, we set out at the beginning in a search for the template that defines the form of the body. And just to sum up, well, we didn't find it in DNA. And we concluded that there was a hierarchy of coordination from higher levels to lower levels within the body, from the level of the organism right down to the use of, the correction and the augmenting of DNA information. We saw that there was no morphological template within DNA, but it may be a factor of the overall organism, not of its bits. And what we're suggesting is that there seems to be evidence that the phenotype or the physical form, the physical characteristics can be modified by psychological interests, by the psychology of a creature. Psychology drives biology. We saw that in the example of E.COLI, the mussels, the stickerbacks, the grooming of the primates, and then the fear in the mice. So what all of this is leading us to is that there seems a possibility that the template for the physical body of a species may be related to the psychological interests, and the source of the psychological interests, the mind, linked to the source of consciousness within that body. Is there a connection between morphology, consciousness, intention? That's where we seem to have reached at this point. And from there, we'll continue our search next week. We won't be taking questions because of the arrangement that we have, but we'd like you to email us with questions to info@s-pi.org. And otherwise, I shall see you next week for 3.7, which is a new model for species morphology. Thank you very much. We'll see you next week.